Good morning, everybody, and thank you to the press for coming to today's press conference. The IASLC Communications Committee, on behalf of the IASLC, is very pleased to have selected the top 20 abstracts from this year's World Conference, and these are the last few from the total of the conference, so thank you to our speakers here today. This morning's press briefing will include 4 abstracts that were judged among these top 20, and they represent every country and nearly every oncology discipline. Just 2 housekeeping notes. Please hold all questions until the end of the briefing, and remember that you can view the press briefing again on the ISLC YouTube channel later today. So from this point of view, we'll start with our first presentation, which is from Dr. Jeffrey Liu from the Princess Margaret Cancer Center. Today, Dr. Liu will present efficacy and safety of taletrectinib in patients with ROS1 positive non-small cell lung cancer, the Global Trust 2 study. Thanks, Jeff.
Thank you very much for the introduction here. So I will be presenting this particular trial, the Global Trust 2 study. These are my disclosures. So Trust 2 is a global study investigating an oral once-a-day next-generation targeting TKI drug called taletrectinib in ROS1 positive advanced stage non-small cell lung cancer patients. So Trust 2 has, in this case, being presented with 2 registrational cohorts. So there are two separate cohorts that are being presented. The first is in the TKI naive group consisting of 55 patients, and the other one is in the TKI pretreated group consisting of 50 patients. What we do know that in this trial is that the baseline demographics and the disease characteristics in this trial are consistent and representative of what we see in the ROS1 non-small cell lung cancer patients. So although it represents a relatively small proportion, approximately 1% of non-small cell lung cancer patients, it does actually impact on tens of thousands of patients worldwide yearly in this setting. And these patients, as you can see in the demographics, tend to be younger, they tend to be fit, they tend to be never smokers, and they also have a reasonably high chance of developing brain metastasis as well too. So taletrectinib was actually treated in the cohort of previously untreated patients. And here, we're able to demonstrate an excellent overall response rate of 85%. The waterfall plots shown on the left indicate that the vast majority of patients actually had shrinkage of tumor, even if it didn't meet a response rate of a partial or complete response. On the right-hand side, you have the intracranial response in the brain itself, and that rate was 67% in terms of response rate. And again, even those individuals who did not meet the definition of a response tended to have shrinkage of tumor as well too. What we also were able to demonstrate was that in TKI pretreated patients, so individuals who had previously received another TKI and then went on to taletrectinib, the overall response rate was 62%, and the intracranial response was also a very good 56% as well. The other important thing about taletrectinib has to do with its safety profile. So taletrectinib demonstrated a favorable safety profile. The most common side effect leading to dose reduction were actually asymptomatic elevated liver enzymes. So patients were actually not complaining about these symptoms. They were a laboratory abnormality that led to the decrease in the dosage. More importantly, or equally as importantly, the rates of neurological adverse events were low. And that's because the class of drugs that targets ROS1 often will have a lot of these side effects. So for dysgeusia, which is a change in the taste, as well as dizziness, they were under 20%, and the vast majority of those were grade 1, some grade 2, and none of them were grade 3. Treatment discontinuation due to adverse events were low at 7.5%. So the take-home message is that taletrectinib is a new oral once-a-day TKI drug for use in patients with metastatic ROS1 lung cancers. And Trust 2 is a phase 2 study with global geographic representation that evaluated both individuals who never received a previous TKI and those who received one prior line of TKI. Both cohorts had excellent confirmed objective response rates and intracranial response rates. Importantly, the drug was very tolerable with very few neurological adverse events and low permanent discontinuation rates. So TRUST-II demonstrates taletrectinib's potential as a highly effective and safe drug for patients with advanced stage ROS1 tumors. Thank you very much. So we were supposed to hold all questions until the very end, but unfortunately, I have to actually present this literally in about 25 minutes down the hall around the corner. So they've allowed me to take the questions now.
Yes, that would be great. Any questions from the audience? And Chris will be able to bring you a microphone. Any questions online? Okay, I think in the absence of questions, Jeff, you're off the hook.
Thank you so much.
We'll field questions afterwards and
Sure.
forward them to you next.
Sure, thank you very much.
Okay, with that in mind, I'm pleased to introduce our next speaker, who's Dr. Jacob Sands from the Dana-Farber Cancer Institute. And today he's going to present the final overall survival data from TROPION Lung-01, datopotamab deruxtecan versus docetaxel in patients with non-small cell lung cancer. Thanks, Jacob.
See if you need help with your slides here. Thank you. So I have all the slides here, but I'll go through them a little more quickly than I did there. First of all, in background, this was a datopotamab deruxtecan versus docetaxel in non-small cell lung cancer. This was previously reported at ESMO. The dual primary endpoints, the median progression-free survival, was positive. So this was already reported as a positive trial, but we had the update for the overall survival analysis. This was a little background just to remind people the intent to treat there on the left, but really it was the non-squamous population that benefited from this drug with an improvement in median progression-free survival and an impressive more than doubling of the overall response rate. Here's the schema from the study. I mentioned it has squamous and non-squamous both. It also had patients who had tumors with and without actionable genomic alterations. Within the study design, what was required in prior therapy is there on the left. In tumors without actionable genomic alterations, patients had to have received platinum-based therapy as well as a checkpoint inhibitor, either concurrent as a single line or sequential as 2 lines of therapy. In those with tumors with actionable genomic alterations, specifically the ones that you see listed there, they had to have received 1 or 2 prior approved targeted therapies as well as platinum-based therapy, but with or without checkpoint inhibitor. They were randomized to Dato-DXd or docetaxel at the doses you see there. And the histology of squam and non-squam, as well as the presence of actionable genomic alterations, were both part of the stratification. In the baseline characteristics, I pointed out in the bottom left there, 78 and 77% of patients enrolled that had non-squamous. That is really the population of most interest given the results I've highlighted. On the top right there is the actionable genomic alterations that were present in 17% of patients. Brain metastases were allowed at baseline if they were treated or if they were untreated and stable asymptomatic. And so both of those are listed within that percentage you see there. Here are the overall survival intent to treat curves. So this is the new data or the updated data. And you can see the medians were 12.9 and 11.8 with hazard ratio of 0.94. Now, we saw the same thing from the progression-free survival curves, with the exception of having a statistically significant difference, a bit more separation. But what I'm getting at is a same thing where these curves looked quite different when we look at squam and non-squam. In the forest plot, we get a hint at that, but I'm going to skip right to the survival curves. And I think this is really the highlight of this presentation is the update for these survival curves. The squam there on the right, which was in favor of docetaxel, and then one on the left where we see a 2.3-month improvement in the median overall survival at 14.6 months relative to 12.3 with a hazard ratio of 0.84. I thought the discussant did a great job in highlighting the historically outcomes of docetaxel, that in this study, the docetaxel arm outperformed historical norms, but 16% risk reduction for death, 2.3-month improvement in the median overall survival in the non-squamous group. We also looked at the bottom there, you see in those with tumors with and without actionable genomic alterations. When one was present, the median overall survival difference was 15.6 as compared to 9.8 months, and in the absence was 13.6 versus 12.3. This was looking at subsequent therapies and how that may have impacted the analysis. Essentially, the top line on this is that the later lines of therapy were not what was causing the overall survival difference. On all accounts, it really seems from the therapy given during the study. As far as a safety profile, with the additional 11 months of follow-up, there was no change in the safety profile. There were no late toxicities observed. We did see that the median treatment duration for Dato-DXd compared to docetaxel was more. It was 4.2 months as compared to 2.8 in the docetaxel arm. And despite that, there were fewer dose reductions at 20% as compared to 30% with docetaxel. There were fewer treatment discontinuations at 8% relative to 12% for treatment-related adverse events. When looking specifically at the different toxicities and symptoms, there was no change to what we've previously seen. Stomatitis, mucositis continues to be the most common in the Dato-DXd arm. This was overwhelmingly grade 1 or 2. In docetaxel, we know that cytopenias are quite common, and we certainly saw that. There were no new drug-related ILD events, so overall, very similar safety profile. So the essential, the big point on this slide, well, I'll make a minor point actually before the big point. The minor point was that we previously saw the TROP2 normalized membrane ratio that was presented at the presidential. This is exploratory and very new. This is really fresh data. So in the progression-free survival, it seems like we may actually have a biomarker based upon the progression-free survival analysis, and there is ongoing testing for that. There will be a lot more data that comes out from that in the months ahead and further advancing what looks like a promising new biomarker. But overall, from the study that we have with the update, we have a drug that when compared to docetaxel in the non-squamous population showed a statistically significant improvement in the median progression-free survival, a more than doubling of the response rate, an improvement in median duration of response, a toxicity profile that I think is complicated but appears favorable, and a median overall survival benefit that matches what we're seeing from the progression-free survival. And with all of that together, highlighted this as a potential new therapeutic option for patients with non-squamous non-small cell lung cancer. Of course, this drug is currently in front of the FDA for review. Thank you.
Thanks so much, Dr. Sand. We will retain questions for the end. So our next presentation is on abstract TZOV in Asian patients with c-Met protein overexpressing non-squamous EGFR wild-type non-small cell lung cancer, results from the Luminosity study. And today this will be presented by Dr. Hidehito Hironuchi from the National Cancer Center Hospital in Japan.
So thank you for your introduction. And I'm Dr. Hidehito Hironuchi from National Cancer Center Hospital, Japan. And also I thank to select this abstract for the press meeting this time. So I'd like to share about the Luminosity trial. This presentation is especially about the Asian subgroup, but I will cover also the overall patient population. Closure. And so I would like to give the summary of the MET expressing non-small cell lung cancer. About 25% of the patients with non-squamous EGFR wild-type non-small cell lung cancer are showing the overexpressing MET status based on the IHC. So another point is the c-MET overexpression is related to the poorer prognosis in these patient population, also in Japan, in Asian races. So Telisov is a first-in-class c-MET targeting antibody drug conjugate, a little bit different from the Dato-DXd. It's all KAMA-based, but we have conducted the phase 2 trial in two-stage design. So this is an overview of the Luminosity trial. In the first stage, we have selected several patient populations, including non-squamous EGFR wild-type, including the non-squamous EGFR mutant and squamous non-small cell lung cancer to select the best population to seek the further explanation. All the patient is eligible with the c-MET overexpression by IHC The definition of the c-Met overexpression here is shown under the figure. The 25% or more membrane expressions in 3+ intensity is defined as a MET positive. And then we have divided these patient population in intermediate and high based on the cutoff of the 50% or more or less. The primary endpoint is objective response, and based on the first stage, we have selected EGFR wild-type non-squamous cohort for the further exploration. And the EGFR mutant is going to the combination of the osimertinib, and the squamous, we haven't selected it for the further exploration. So this is a baseline demographics. So I think that many of you do not have the information about the c-Met overexpressed patient population in non-small cell lung cancer. I can disclose it in the median age was 64.0-year-old, and the majority of the patients have the smoking history, and brain metastasis found in the 20.5% of the patient. Other characteristics is listed in this table. And this is an overview of the overall study of the luminosity trial. When we check the objective response in the left upper side, the red bar shows the overall response rate based for the patient with c-Met overexpressing population. The response rate is 28.6%. And when we select these patient population by the c-Met expression, the high subgroup shows a 34.6%. Of note, this patient is in the second and third line patient population. The usual standard of care for about the docetaxel shows only around 50% of the response rate in this patient population. And also we check the duration of response, and the right side of the table, you can find the 3 months of the median duration of response, and notably the 56.5% of the patient enjoys more than 6 months duration of response. And again, this is a c-Met overexpressing patient population who usually reported as a poor prognosis. About the adverse events, safety profile, the most common safety events is related to the peripheral sensory neuropathy. Grade 3 or more has been reported in 7.0%, and also the important one is the drug-related ILD. The retrospectively adjudicated drug-related ILD was observed in 9.9% of the patient, and there is a 1.7% of the grade 3 in 3 patients. However, the 2 of the 3 adjudicated deaths were considered directly related to progression of disease by investigator. And when we check the peripheral sensory neuropathy, 10% of the patient has discontinued the treatment based on the TELISO-B peripheral sensory neuropathy. However, the median time to the onset of the peripheral neuropathy leading to discontinuation was more than 200 days. It means the majority of the peripheral sensory neuropathy related to the discontinuation happens in the patient who experience the efficacy of this drug. And this is when I would like to get into the Asian subgroup, but the patient characteristic is quite similar for the overall patient population, and also that there is a patient disposition in message, but I will skip this slide. And this is an efficacy overview of this trial in Asian subgroup. As shown in the previous slide, the red bar is the objective response in the overall patient population with c-Met overexpression. As usually, the Asian subgroups show something like the better response rate or better efficacy, and in this figure, the response rate in the overall patient population is overexpression is 35.4%. And when we select the patient with MET high status, the objective response rate increases as high as 46.2%. The duration of response is similar to the overall patient population, around 7 months, and the half of the patient enjoys more than 6 months of the duration of response. When we check the PFS in the left side, the median PFS is 5.5 months, and there is no difference between the high and the intermediate patient population. And when we check the overall survival, there is a numerically better overall survival trend in CMET high patient population. That was a 25.4. But when we check the overall survival curve itself, there is not so huge difference. Maybe this is why, based on the small sample size in this subgroup analysis. So this is a safety overview. As like the overall patient population, the peripheral sensory neuropathy based on the MMAE is the highest number, but only 1.8% of the patient experience grade 3 or more. And I would like to show the ILD in this patient population. Also, the retrospectively adjudicated drug-related ILD was observed in 15.8%, a relatively high, but it's also usually in the Asian subgroup, and 8.8% has the percent of the patient have the grade 3 or more event. And there is a 3.5% of drug-related deaths in grade 5 happened in this analysis. And similarly, the previous safety signal, one of the 2 adjudicated deaths was directly related to progressive disease by investigator. In conclusion, treatment with Teliso-V resulted in compelling and durable response even in the patient with CMET overexpressed, usually poor prognostic patient population, especially those with high CMET status. These results are comparable with the overall patient population. And about the Teliso-V, there is an acceptable and manageable safety profile, and especially the sensory peripheral neuropathy occurs in the late on after the efficacy signal happens. We are now doing the TeleMet non-small cell lung cancer 01 trial. It's directly comparing the Teliso-V versus docetaxel in the same patient population. Stay tuned. Thank you for your attention.
Okay, so that takes us to our last presentation today. We're very excited to welcome Dr. Chen from the British Columbia Cancer Research Institute to present an abstract entitled Exposure Impact on Never Smoking Lung Cancer Patients. Thanks very much.
Thanks for the introduction. So good morning, everyone. I'm Dr. Yishan Crystal Chen, a researcher at BC Cancer Research Institute in Canada. I want to thank the WCLC organizers for selecting our abstract. I also want to thank Dr. Stephen Lam and Dr. Rafael Meza for their mentorship. Now let's get started on how exposure to fine particulate matter affects lung cancer patients who have never smoked. In 2013, the International Agency for Research on Cancer classified outdoor air pollution as a group 1 carcinogen, meaning it causes cancer. One of the most concerning parts of air pollution is fine particulate matter, also known as PM2.5, which consists of tiny particles that are 2.5 microns or smaller in diameter. These small particles can go deep into the lungs and even get into the bloodstream, posing serious health risks. For people who don't smoke, PM2.5 is a major risk factor for lung cancer, especially in females. However, we still don't know the most critical timing and the duration of exposure that are clinically relevant to lung cancer. Our study looked at how both short-term and long-term exposure to PM2.5 could affect genomic changes in lung cancer patients who have never smoked. Our study involved participants from the BC Cancer Vancouver Center. Each participant provided their residential history, which we used to estimate their exposure to PM2.5. We did this by geocoding their addresses and combining high-resolution data from satellites, chemical transport models, and ground measurements. We focused specifically on genomic variables such as EGFR and ALK mutations to see how this might be impacted by cumulative PM2.5 exposure. To analyze this, we used logistical regression. Our study included 255 patients with no EGFR and ALK statuses, with 56% being EGFR or ALK positive. We found significant association between EGFR status and patient characteristics. EGFR positive patients were more likely to be Asian, born outside of Canada, diagnosed at stage 4, and to have adenocarcinoma. In the 188 female patients, we observed a trend of higher cumulative exposure in the EGFR positive group. The cumulative 3-year exposure was 19.0 micrograms per cubic meter in the positive group versus 17.3 units in the negative group, with a p-value of 0.031. For the 10-year exposure, it was 78.6 units in the positive group compared to 69 units in the negative group, with a p-value of 0.219. In our analysis, we found short-term exposure to PM2.5 is especially important. The odds ratios are around 1.2 to 1.3, which shows a substantial effect size. Even though the confidence intervals are wide, this still suggests a strong link, especially in the crude models. This means short-term PM2.5 exposure could play a key role in promoting lung cancer in people who don't smoke, particularly through EGFR mutations. On the other hand, for long-term exposure, the confidence intervals are much narrow, meaning our estimates are more precise. But since all these intervals include an odds ratio of 1, there isn't a significant association with long-term exposure. So it seems the risk of lung cancer with EGFR positive status from PM2.5 is more noticeable with short-term exposure rather than long-term, especially for non-smokers. In conclusion, our study suggests short-term cumulative exposure may impact genomic alterations in female lung cancer patients who have never smoked. While the crude model shows a significant association, the adjusted model indicates a trend that, although not significant, warrants further study. The prevalence of adenocarcinoma among patients with genomic alterations also highlights the importance of understanding these specific changes. Looking ahead, we need to confirm these results with larger cohorts and in different geographical regions. Additionally, we would like to explore more covariates, which will also improve how we measure exposure. This will help us get a clear picture of the mechanisms at play. This research opens important avenues for understanding the link between air pollution and lung cancer. Finally, this study was funded by Terra Fox Research Institute, BC Cancer Foundation, and the VGH and the UBC Hospital Foundation. Thanks for listening.
Thanks so much, Dr. Chen. Really fascinating results. And to conclude, and before our discussion, very pleased to welcome Janet Freeman-Daly, our ISLAC patient advocate up. She is going to provide a unique patient perspective on the research presented this morning. Many of you will have had the pleasure of working with Janet as one of our institutional and professional society patient advocates. She's well known for hosting the Lung Cancer Social Media Chat, is a patient research advocate and co-founder of the Ross Wonders, a former associate editor of the ISLAC Lung Cancer News, and co-developer of the ISLAC flagship STARS program. So Janet, very pleased to have you today and welcome you to the stage. Thank you.
I'm a person living with metastatic lung cancer. You probably haven't met many of us. For decades, there were few lung cancer patients who survived to be met. Only 15 years ago, the majority of patients died within a year of diagnosis. But thanks to research like what was discussed so far this session, the number of long-term survivors is growing. In the US alone, there's over 650,000 people living with lung cancer. I was diagnosed in 2011. My cancer progressed rapidly despite 2 different lines of chemotherapy and radiation. My oncologist told me I would probably be on chemotherapy for the rest of my life, which would likely be short. But I was very fortunate. I was able to access a new biomarker test for a gene alteration called ROS1, and I entered a clinical trial for a new targeted therapy pill. I have been on the same drug for almost 12 years with no evidence of disease. I'm not considered cured, but scans can no longer detect cancer in my body. However, there may be some undetectable cancer there, so I continue to take my pill and will for the rest of my life. But I have a good quality of life. What a change in just 15 years. Other people diagnosed with lung cancer, however, are not so fortunate. I'm a co-founder of the ROS Wonders, a group of patients and caregivers living with ROS1 positive cancer. While we have many members who have survived 5 or even 10 years, like me, on targeted therapies, we also had many members who ran out of time. Their cancer treatments either stopped working due to acquired resistance, or in some cases, didn't work at all. We've even had some people who ended up finding they were diagnosed with the wrong biomarker. In others, the cancer spread to the brain, which some of our drugs don't treat. That's why we need research. Some of the research discussed today relates directly to my type of cancer. The Global Trust II study discussed a targeted therapy for ROS1 positive non-small cell lung cancer, and the data seems to indicate it will overcome some of the resistance mechanisms that people develop on other ROS1 targeted therapies. It also treats cancer in the brain. This is a huge area of need, since over 60% of the patients with ROS1 positive cancer end up with brain mets. Members of the ROS Wonder are grateful that we may soon have another treatment option that can help us live longer with good quality of life. Patients on all types of lung cancer treatments can develop resistance to therapy. The Luminosity and Tropion trials are studying a different type of treatment called antibody-drug conjugates. People with lung cancer are hopeful that newer drugs like ADCs may someday provide a new treatment option for those who develop acquired resistance to several types of lung cancer treatments. You'll notice many of these clinical trials discussed focused on endpoints that measure survival in some way. While increasing the time that people live with lung cancer is definitely worthwhile, other measures are also important to patients. People who are hopeful of being cured of their cancer might be willing to put up with considerable side effects for a few months. But those like me who are kept alive by ongoing treatment have a different perspective. Those of us with metastatic disease do want to live longer, but not at the cost of being miserable all the time. We need to know what it's like to be on a treatment so we can make good decisions about how we want to go forward. We need to know how that treatment will affect our ability to function every day. And while the physicians do measure side effects, it's from the physician viewpoint. They can't know how it feels to be on this drug and to function. We'd like to see trials include more patient-reported outcome measures to capture what it's like to be on a treatment and to have options for dose reduction when side effects become challenging. We'd also like to know more about the risk factors for developing lung cancer. The fact that so many people with no known risk factors like me still develop lung cancer tells us we have a lot of work to do in this area. I'm glad to see the study on the PM25 exposure impact on people who develop lung cancer despite having no smoking history. As the climate changes, we're having more wildfires, which generate those particles and can create air pollution thousands of miles away. People who have a family history of lung cancer or who have other environmental exposures, including air pollution, need a better understanding of their lung cancer risk. Lung cancer screening needs evidence to allow us to include people who have a wider variety of risk factors. Anyone with lungs can get lung cancer. As a patient research advocate, I work with researchers to identify research questions that will address areas of highest need for patients and hopefully improve outcomes. I work with the people who are doing the Trust II study, and I work with several others. I appreciate the researchers who include patient perspective and co-develop research projects with patients as partners. Let's keep research moving forward so that people who get lung cancer can have a normal lifespan with good quality of life. Thank you.
Thanks very much for that very poignant summary. I think it really places all the data into context. From here, Chris, I think, is going to moderate some of the questions that come from the audience, some of the questions that come online, and the panel. Thanks very much.
Any questions online? No. Okay. That concludes the briefing. Thank you so much for coming.